Good evening, ma'am.
Good evening. How are you?
I'm fine, ma'am. How are you?
Yeah, I'm good. So we'll begin the today's session, and today's session is on single cell genomics and small RNA analysis. As the name itself indicates that we are going to talk about single cell genomics. So whatever the nucleic acid contains, whatever the genomic aspects are present in single cell, we are going to look at it. As well as we will look into brief what are the small RNA kind of analysis we could do. Because last in our previous sessions we have conducted a transcriptomic, but we didn't cover small RNA. As today we are going to see single cell, and it is more or less the similar kind of analysis. So we will cover small RNA analysis as well. So in today's session we will be covering the introduction, single cell genomics, workflows which can go for single cell and small RNA, library preparation, as well as data analysis, how to generate the data analysis, various kind of applications, and we will summarize with some hands-on session. So let's begin for today. So the human body consists of 37 trillion single cells represented by over 50 organs, which are stitched together to make us who we are. Yet we still have very little understanding about the basic unit of our body. What cell types and states make up our organ, both compositionally and spatially. Now, with the advancement in genomic technologies, the profiling of the human body at a single cell resolution, it is possible for us to generate an unprecedented wealth of the data, which will accelerate the basic and clinical research to understand the biology of gene expression and gene variations, and which can give us the leverage of to the future medicine. To date, several major organs have been profiled at a single cell level, but the challenges lies in various ways to integrate how to integrate the single cell genomics in a meaningful data, so that we can understand the biology of our body and the functioning of different type of cells. In recent years also, there have several consortiums began to introduce harmonization and equity in data collection and analysis. So single cell sequencing technology is transformative to profile genetic, epigenetic, spatial proteomic, and lineage information in an individual cell. It provides unique opportunity, and alongside computational challenges are there for integrative methods which can jointly learn across multiple type of data sets, like genomic, RNA, ChIP-seq, methylation. These kind of data, if we integrate together, then we will be able to understand the biology and the functions of the gene in a much better way. The integrated analysis can discover relationship across cellular modalities, learn a holistic representation of the cell state, which could enable the pooling of the data sets and produced across individuals and technology platforms. So let us see what are the organs and the cells which has been profiled at single cell level. So here in the table, if you see that the cells from blood, brain, kidney, lung, and there are many organs which have already been sequenced at a single cell level for various diseases. And these are the representation of the system. So using these single cell genomic data, because these are a representation from the different autoimmune disorder, immune disorders, and carcinomas. So this makes us this kind of data gives us the opportunity to understand the clonal representation in each disease, because these are the complex disease. The single cell sequencing examine the sequence information from individual cells, which optimized the NGS technology. It provides a higher resolution of cellular and differences and a better understanding of the function of an individual cell in its microenvironment. As we understand that for the immune disorders, cancer, the microenvironment is very complex and multiple genes, proteins at different time points are involved. So this gives us the opportunity to learn each and everything. So to make this technology successful, there are many consortiums which have been established over the period of time. So let us look what are the different consortiums has been established to achieve this goal. So in this table, if you look, these are the consortium. It is just the table. So this table is growing day by day. So and these are the technology where we can achieve these are the platform, these are the academic institutions. They perform single cell, they do the qc, they collect the data from different sources, do the qc and deposit in a, so they process, pre-process, and then they take the information from all these kind of data sets. So now how we can integrate and generate different kind of data from the single cell. So if we look at this figure, we can integrate the DNA technologies, not only RNA. We can do the DNA, we can do whole exome, we can do whole genome, we can do methylation analysis, we can do chromatin instability, and so on. So here you can look in the bold, it is mentioned the technique which we can do, and then below what are the methods already available and establish the protocol to generate the data. And there are many bioinformatics pipelines also available to integrate these entire data together. As we can see, the data is from single cell, but from one sample itself, we are typing thousands and millions of cells. And from each cell, there is a lot of huge data coming out. So the integration of each and every data type is a challenge, it's a challenge, but over the period of time, many more analysis pipelines are coming out. So let us see what are the experimental workflow to generate the single cells and data from the single cells. So if we look at the workflow, there are the different chemistries by which the libraries are generated, whether it's a targeted read depth or UMI barcode amplification or feature. We use features like CD markers and so on. So this is a CD markers. Features are the CD markers. Like we take either the CD44 positive cells, 34 cells. We select using the fact sorting and then perform the library preparation and so on to generate the data. So these are the different kind of platforms. Library preparation kits are available for different kind of platforms. If we look at this particular diagram, it states that how the different kind of analysis we can generate single cell epigenetics, we can see DNA sequencing, we can do the T cell receptor sequencing, we can do the RNA sequencing from the tumor microenvironment. Any kind of microenvironment, whether it's a tumor or any genetic disease which have clonal expansion, drug resistance, microbe or host pathogen interaction, host microbe, antimicrobes, or antimicrobial resistance. So because we have different kind of cells in these environments, so we can utilize the power of RNA sequencing, T cell receptor, or the DNA sequencing and epigenetic analysis, and we can integrate together to understand the tumor biology or the microenvironment biology. So this figure I will elaborate in my next slide. This is basically just the representation how we take the single cell, we lyse it in the tube, and then we perform the different kind of chemistry to generate either the epigenetic data or the RNA data or DNA data or any other kind of data. So this library preparation is depending on the aim and objective, which kind of data we would like to generate. And then when we have the library preparation ready, then we go for the sequencing and generate the sequencing data, and then the downstream analysis is being performed on that. So let us look in detail how the library is prepared for single cell, because that is very crucial. Because why it is crucial? We are not taking the entire nucleic acid or the entire DNA or RNA in this. As a first step, we are doing the cell separation. So we are making the single cell suspension. So there are different techniques to make the single cell suspension. Either we are doing the limiting dilution method. So in this, what we do, we make the serial dilution, and we assume that at some time point we are having only single cell in our suspension. So by limiting dilution factor, we dilute the cells, and then once we arrive at the single cell suspension, we take that and go for the library preparation. Another method is the micromanipulation, which involves the collection of single cells using the microscope guided capillary pipettes. Say suppose if we are doing the culture of any cells, so we observe the plate under microscope and just we pick one colony or single cell out of one colony or from different colonies and so on using the pipette tip. In the third method, we use the FACS sorter. So in FACS sorter, we just first we make the cell suspension and then we pass through a nozzle and then we collect the single cells in different tubes. So that is a FACS sorting method. Another method is a laser capture microdissection. This is being performed on the solid samples, tissue samples, because when we have the slides or when we have the FFP block, we cut the sections, put on the slide, and using the laser, we just pick up one cell out of it. So this is how usually the people or the labs which are working on tumor tissues or they have the FFP block, how they take the single cells out of it. Because this is more precise instead of just first making the cell suspension. So in this, if the tumor tissue itself is very small there, it is very difficult to take that tissue, cut tissue, and then from there, again, we are taking the single cells and losing a lot of the cells which are of our interest. So using this LCM microscope, then we are just picking up only a single cell from the tumors, not from any other area. The another method is the microfluidic technology for single cell isolation. Is require nanoliters volume, that's it, not more than that. So in this, again, we pass this cell suspension through a nozzle, and then it separates only a single cell with a single drop. So it captures only a single cell in each drop. So it's a nanoliter volume. So this is the technique is called oil suspension microfluidic approach. So this is more precise and now more common for the single cell genomics. Once we have the single cells, then now we have to choose which cells are of our interest. So because from each cell we generate huge amount of data, but as we know that it's a single cell, we can't generate from each cell. We cannot generate many more reads. So we have to enrich our cells. So using a magnetic bead technology, each bead will be attached with the antibody, specific antibody of interest, and then we enrich the CTCs. And then on these CTCs, the PCR will be performed. And then from in each drop itself, the PCR will be performed and the library will be prepared, and then these libraries will be sequenced. So now this is the sequence. Now we have seen the library preparation. So these are the different methods, and we will see how specific methods can be utilized because they are not different. Just this particular approach is different in single cell genomics. Otherwise, if you would like to do a whole exome, whole genome, or ChIP-seq methylation, or any kind of data generation from these cells, the library preparation remains the same and the approach will remain the same as the we do from the either the blood sample or the tissues or whole tissue itself. So now let us see, now we have the cells. So basically we have these cells, different kind of cells from the same samples, and each cell will have a different kind of data. So how we identify which kind of data is respective to each cell. So let us have a look at this. So in this, we can see very well when we get the reads, it will be a mix of the reads. But what is the beauty of this single cell genomics? Each read will be having the barcode in it. So say suppose I am taking the blood sample. In my blood, we have different kind of cells, like we have RBCs, we have monocytes, we have eosinophils, and we have lymphocytes and so on. We have different kind of cells. So now when we have single cells, so each cell we are barcoding it. with the different barcodes. So these barcodes can identify each type of cells. So we get the data like this, and then in the second step, we cluster the reads depending on the cell types. So we can see here the different kind of reads are there. And in the second step, we cluster the reads. So if you can see, the red color is of one type, blue color is of one type. So this kind of a clustering is being performed, and from this kind of clustering we make the matrix. So now you can imagine, from one sample, say suppose if we have 10 type of cells, so we get the 10 clusters, and in each cluster we get some thousands to million reads. So how huge the data will be. And in one experiment, we are not just considering only one sample, 10 cells. So we will be having multiple samples like this. So say suppose if we say that 5 samples, 10 cells, and in each 10 cells we will have million of reads. So the analysis itself is very crucial, because we have to do the normalization, we have to do the dimensionality reduction to reduce the data and make the more crisp data. And then if you will look here, then this you can see clearly these are the cell cluster in this. So these are the clusters from one sample. So you can see these clusters and how many dots are there in each cluster. That many number of samples and the cells will be there from each sample. So you can see that this is one sample, this is one sample. We will see all these things in detail. So now we can integrate this genomic data with the imaging also. So from flow cytometer or cytometry data, we get the imaging of these cells. So these images also can be integrated with this genomic data. So now we know these are the cells, cell type, different cell types, because that is categorized based on the genes expressed on these cells. So there are some basic genes we already know what genes are categorize these cells into different cell types. And in cytometry also, or the IHC or histopathology, we know which type of cells they are. And this data is easy to integrate with this data and then make more sense of it. So this is like now more approachable approach for single cell genomics. So now there are different kind of application. As I said before, we can do any kind of analysis. So on the single cells, we can do whole exome analysis. So why we have to do a whole exome from single cell, and how it will help us to understand. So why we are doing the whole exome? Say suppose we have some tumor in it, but this is a small tumor. So if we take the tumor cells and we take the blood and we do the whole exome analysis. So whole exome means only the genes which will be coded, or which will code for the proteins, that those genes only we will be able to see. So if we integrate this information, blood information, because from the blood we will get the germline mutations, and from the tumor samples we will get the somatic mutation profile. So if we integrate the germline information with the somatic information, we will get to know how the genes are getting changed, whether it is coming from the germ cells itself, or it is only in the somatic cells. So this information will help us to understand the mutation spectrum of the tumor microenvironment and help us to design the treatment for the patient. Because the treatment will be different if the mutations are germline, and the treatment will be different if the mutations are somatic. So if we do this, so we can take the single cells from both the samples and process, and then we will get the complete analysis how these two informations are important in the treatment designing and disease association, how the disease will progress further. Second, most important application is the clonal heterogeneity. As we know, in the tumor any kind of tumor, whether it's ovarian cancer, colon cancer, or breast cancer, so we know that already the immune infiltrating cells will be there, which gives us the clonality of the tumor. So this is the unique phenomena of the tumor microenvironment. So if we take the single cells, many multiple cells, thousands and thousands of cells from the one tumor microenvironment, so we will be able to understand which kind of clones are there and which clones are not there, and how these clones are getting mutated, so that the treatment is also getting altered. And in leukemic stem cells, if we map the each and every clone, so we will be able to map the hematopoietic stem cells clones. And then say suppose if monocytes which gives uh, these stem cells, they will become mature and say produce different kind of cells in our blood. If any stem cells is not producing, so it can be taken care of. And this also gives us the information about the how the tumor heterogeneity provides the resistance towards the drug. So as we see that here in the lung mass, we have different kind of clones present. So maybe these particular clones are sensitive to drug, but maybe this particular clones are resistant to drug. So if we could map these different kind of clones, different time point, so maybe we can help to make them expansion, which are the normal clone, by destroying only the other type of clones, so that we will be able to overcome the problem of drug resistance. Cell lineage and the liquid biopsy. If I talk about the cell lineage, so we know there are different developmental stage in the body, like oocyte, fertilized egg, medulla, blastocyst, and then it will turn into the mature cells like a neuron cells or any kind of other cells. So if we have, because here we have only one cell, two cells to get these opportunities, right? So if we understand how what are the transcription factors are available which makes the genes to express, then it will be easy to understand the developmental biology. And in liquid biopsy, this provides an exciting opportunity and single cell RNA-seq of CTCs, which would provide novel insights into biomarker characterization. The intratumor heterogeneity poses challenges in cancer genomics. Single cell RNA-seq can tackle this problem by effectively identifying the subgroups based on the responsiveness in various contexts, whether it is mutated, not mutated, whether it will be drug sensitive or drug resistance. So all these phenomena, it is possible with the help of single cell genomic data. So if we look at the lineage, as I already told to you, that it can also identify the novel differential markers. Now, as I mentioned that from the single cell, we can perform any kind of genomic data analysis, whether it's a DNA. From the single cell, we can isolate the DNA, we can isolate RNA, we can isolate protein and do the integrative analysis. So let us have a look and in detail about the single cell RNA sequencing, because if we see the general phenomena, if we have the RNA sequencing data with us, we can either predict the protein or we can go back and check the gene information as well. So if we look in here, then first the single cell isolation, then sequencing data, quality control, read mapping, normalization, then the different kind of analysis we can do. So first is the population identification, which kind of different kind of cells, clones are present, differentially expressed genes, then these cells, what are the different kind of cells and their population size, that is called the cell trajectory analysis. Then the network reconstruction. So from each cell, what are the genes, what are the proteins will be there, so we can create a network between them. Then we can integrate this single cell data with the whole transcriptomic data and can perform other type of analysis like alternative splicing, allele expression, RNA editing, and so on. So, and here in this table, there are different kind of methods are available to generate the transcript, single cell transcript data. So if we look at these first few couple of library preparation methods and the platforms, they gives the full length. But if we look at the below methods, they gives either the 5' or 3'. It means that they generate only a single end libraries. But here it gives the paired end so that it will give the full length single cell transcriptomic data. So now let us see what are the different analytical steps and the quantification methods. So the single cell sequencing data are inherently noisy because it will have so many other things as well. Because it will sometimes what will happen, you won't get, if you again it depends on your how good is your single cell isolation method. If that is very good, you will get a crisp data. But if at that time point, if it is not like say you won't be having single cell, say suppose if you are using the limiting dilution factor method, then it might give you more noise. And also because you are taking only single cell and low number of reads as compared to the whole data, it will be much more noisy. So after sequencing, we have to correct for batch effect, then the cell specific capture efficiency and dropouts. Because sometimes what will happen, you in single cell, if you have a mixed doublets kind of a thing, then maybe you are not getting only single cell, maybe it's a mix of the population. So if say suppose I would like to do the only NK cell sequencing, so sometimes when we separate the NK cells from the blood, it might get contaminated with the other kind of cell types, and hence the data will also be contaminated. So after sequencing, the alignment will be the first step, and then deduplications, then the initial gene expression profile matrix, and then the normalization is performed with the raw expression data using the various statistical methods. So every step we perform the additional QC methods to make the data more clean and clear. And these are the different kind of quantification methods one can use. So one is the RPKM, another is FPKM, and third is the TPM. So now these days TPM method is more common than the other two methods. So the RPKM is the read per kilobase, which is identified by the multiplying the read counts of an isoform by 1000 and dividing by isoform length. So that is known as the RPKM method. So TPM is only the while the TPM is based on the transcript per million. So when we make the transcript and then divide by 1000. So now this method is becoming more and more common to calculate the quantification for our transcripts. If we look at the different tools and software for different steps, so as I mentioned, the first step is the alignment. So for the alignment, there are different tools. They are not different. So they are not different, but they are not different. But these aligners are the same as we do it for the normal transcriptomic data analysis. So very popular is top hat, then star, high set. So depending on our aim and objective, we can choose any aligner. There is no restriction to align our reads to the reference. But this we use for the reference based. And usually when we do the single cell genomics, Usually we prefer to have the reference. But in many instances, now people are moving where the reference is not known, and they do the single cell genomics from as a de novo assembly as well, to understand more and more biology from the different tissue types and individual environment within the tissue itself. So after we have the alignment is done, map reads are file are generated. Then the second is the quantification methods. So these are the different methods or the different tools which can be used for the quantification. And these are the tools which we use for the differential expression of the genes. So there is nothing much difference from the normal RNA sequencing data analysis. It is just the data how we have to put together. There we generate only one matrix for each sample. Here the different count matrix will be generated from each type of cells within the sample and then merged together. So there is a very one popular company called 10x Genomics. 10x Genomics is the leader in the single cell analysis. So the workflow from the single cell suspension, they have the 8-channel microfluidic chip, which allows up to 8 samples to be processed in one go. The samples are barcoded individually for a multiplex sequencing reaction. So it means that for each sample, we'll have multiplex barcoding within the sample itself and between the samples. So the 10x Chromium is the instrument on which the sequencing is being performed. And this instrument has the capability of capturing 100 to 10,000 cells per sample and up to 80,000 samples per chip. So you can estimate now how many cells can be sequenced together from different samples and within the sample, which recovers up to 65% of the cells depending on the cell type. The lower doublet rates. So this particular platform has a 0.8% per 1,000 cells. So this 10x Chromium instrument has the capability to separate the cells. So if you look at the workflow on this particular platform, so what it does, it has the a chip like this. It has 1, 2, 3, 4 rows, and on the upper top row, it is filled with the oil, then the cells and the reagent, and then the beads. So which are specific to the cells. So then so the oil will mix with the cells and reagent, and it will travel through the beads, and then it will form an emulsion. And then this emulsion will generate the drop like this. So in this particular tube, so this tube represents one cell. So it has all these beads, barcoded primers, your cells, and the oil. And when it passes through this nozzle, so then it will generate the drops, and in each drop, we will have the cells, reagent, and the beads. And then the beads will be separated based on the antibodies which are coated on the beads. And then on this each cell itself, the RNA will be converted into the cDNA, and then the library preparation will be performed on that. And once the library is performed, it will go for the sequencing and generate the data, and we will get the FASTQ reads from the sequencing data. This is just the representation of these different kind of cells. So this different type of cells is from the single sample. So don't think that this is a different sample. So different color, different sample. No, this is a cluster of cells from the one sample only. And you will have the million of such cells from different type of samples. So this is from the 10x Genomics. They have the end-to-end solution. They have the bioinformatics pipeline as well. I'll show you some of the important steps and the visualization from 10x Genomics, which are open source. So the other pipeline, very common pipeline, is Sincera, which is which is integrated and work with R Studio. And this pipeline, what it will do, it takes this your aligned reads and the count matrix from the aligned reads, and then it process. So automatically it process, it has all these scripts in it. It will do the pre-filtering on based on the expression and the specificity. Then it will normalize the count matrix based on the Z score and the trimmed mean. Then it will perform the quality control using the correlation matrix. And then the second step is the cell type identification using the cell cluster, then the differentially expressed gene from the cell clusters and the cell types. Then it will make the again another matrix from each cell, what are the highly expressed cells or the down-regulated genes. And this is the how it goes like the workflow. So this entire pipeline, if somebody has the expertise on R, they can run on R. So now there, as we have seen before, there are some confounding factors in the single cell genomics. So the technical batch effects is a well-known problem in single cell RNA-seq when the experiment is conducted in different plates. Say suppose I have my cells in different plates from the same samples. So it will have a batch effect because the always the number of cells will be different, and thus the sequencing data generation will also be different. And say suppose if we have one sample in one plate and the replicate is another plate, then again it will give the batch effect. Same is for the different samples if we are processing at same day or same time or at a different time. The cell specific scaling factors, as I mentioned before, that it is not always true that your cell population what you get from the same sample, it is always the same. So we have to normalize from the, and each cell will have their own RT efficiency. RT efficiency means conversion of RNA to cDNA. So that also will be different, and it will also affect the data. And then some dropouts will be there during the amplification or the library preparation, dilution factors, and the amount of sequencing we generate. So every step we consider, we have to do the normalization before proceeding to the second step. The single cell latent variable model can effectively, so there is a model called SCLVM. So this is a latent variable model. If it is applied, so it can effectively remove the variation explained by the cell cycle effect. So we know that even though if we are taking the one cell population from one sample, but every sample will be at different stages of its life. So we cannot say that, okay, which stage it is, because here we are not culturing it. We are just taking the sample and isolating the cells. So they will be at a different time point of the cell cycle. So how to remove this effect? So this is the model which can be used to remove the cell cycle effect. Now the expression value Y can be modeled as a linear combination of R, that is a technical and biological factors, and K latent factors with a noisy matrix. If we get too much of noise, then we have to factor it with the technical and biological replicates. So in our data, we should always have the technical and biological replicates. So now how do we identify? So second step is very important because we have to identify these cells correctly, because the cells are living in a dynamic context which are interacting with their surrounding environment. So the PCA, so that is a principal component analysis, could be used very effectively to identify known and unknown cell cluster. So what PCA does actually, so it forms the cluster and which could be identified in Y and X direction, and in each cluster a different kind of cell types will be differentiated. So to identify the cell types, PCA is performed. Cell hierarchy reconstruction can be performed after 2D projection of the normalized gene expression profile and then mapped to the regulatory network. So now let us see what are the different methods for the cell typing. So if we would like to do the cell differentiation, there are different methods. So TSNE is there, IC and MIST there, PCA, K means MST, PCA diffusion map, and bootstrap with the K means. So these are the different algorithms which can be applied for the different kind of data to identify the different kind of cell types and do the dimensional reduction. So in T-SINE, the main strategy is the principal curve. In IC and MST, it's a differential expression. In PCA, K-means, as I mentioned, the PCAs will cluster the cells in different groups and give us the cells in different dimensions. The PCA diffusion map and the K-mean is the assembly method. So these are the methods. This is nothing but these are the methods. We will have the different kind of equations. If somebody has from the math background or statistic, they understand better on which equations and how they work better. But for a biologist, we understand only, okay, this is the method, this is the algorithm, and how they work it. That's it. Whatever the required input, it is none, just on the, you feed your data, and then the environment is the MATLAB. ICA MIST you can do in R. PCA K-means can be done in R. And PCA diffusion also can be done in Python. So here R and Python is very, very, very important when we create the PCA, volcano plot, heat maps, and clustering of the cells. So one should have the basic understanding of R and Python. So that is the basic requirement for this particular single cell genomics. Though now many web servers, free web servers are available. I'll show you in the hands-on how we can use those servers also. There we can upload our data, as well as inbuilt data is also there, and which can be integrated with our data as a metadata. So now if I would like to summarize the single cell genomics, I would like to summarize that it is a very powerful technique to identify different type of cells, gene expression, and different type of clones. It helps us in identifying the new biomarkers, which could be very helpful in disease identification, disease monitoring, and treatment, identification of new treatment modalities. It is very robust, and robust transcriptomic analysis down to single cell inputs level for high quality samples. So it means that our sample preparation, or the sample which we consider, that needs to be very high quality. Otherwise, this will be very challenging, even for the data generation as well as for the analysis. The integrated protocol proceeds directly for the whole cells and preserve the sample integrated. Say, suppose if we have some data analysis, data from the single cells, so we can assume that the whole entire population can be of the same type. The high resolution analysis enables the discovery of cellular differences, usually which are masked by the bulk sampling methods. So what will happen when we have the single cell? So we know each cell type, what it is, what is the genomic information it gathers. But sometimes what will happen if we type the entire tissue, the clones which are of less number, they will be masked by the other clones. So that is the limitation, and that is the privilege of single cell genomics, which will leverage this information of the rare clones. I could say that the rare clones, or the clones which are in less number. So let us move to the small RNA analysis. So as we know that in the cells, there are different kind of genomic information. So the genome, the transcriptome, and transcriptome is also of two type, is which codes directly for the proteins, that is the mRNA, and more than 80% is the non-coding. Under non-coding, we have the small non-coding, which are the microRNA, and the long non-coding RNAs, which are the LNC RNA. And if we would like to differentiate between small and the long, so small non-coding RNAs are the less than 200 nucleotide, and the long ones are the more than 200 nucleotide. They are transcribed by the RNA polymerase II. They are also transcribed by the RNA polymerase II. They are produced endogenously by the endonucleases. while the long non-coding RNAs are produced mostly by 5-cap polyadenylate and the spliced from the spliced mRNA. They are well conserved, they are poorly conserved. And till date, more than 2000 microRNAs are known and well established, while more than 30,000 non-coding RNAs are known. So I'm not going into library preparation, but I just brief you how the library preparation is done. So during the first we isolate the RNA, so that is a total RNA. So during the RNA preparation also we can enrich the small RNA and just get rid of the long coding RNA, that is the messenger RNA, and then we will left over with the small RNA, micro RNA, that will have your micro RNA, pi RNA, siRNA, all these kind of RNA comes in the your non-coding RNA. So if we are doing the enrichment, poly A tail enrichment, then we will get the mRNA, and the other fraction we can use for non-coding RNA. Otherwise, we can do the ribo minus. If we do the ribo minus, we will have all other RNA except the ribosomal RNA. So this is kind of different kind of library preparation we can perform. And there are, in today's, there are some specific library kits are available where you can just prepare a small RNA and sequence that. Even there are some kits available which by which you will get only either microRNA or siRNA. So we take the samples, prepare the small RNA libraries, and then sequenced. Then after we get the sequencing data, first the adapter removal and get the good high quality reads. Then align to the reference. So because I took the example of humans, so I put the HG19. But if we are taking the plants, so we map all these microRNAs to the reference plant. Then we use the different kind of bow tie aligner. So we can do the bow tie aligner. We can use the bow tie, because bow tie aligner is for the small short reads. So we can use bow tie 1 or bow tie 2. Or otherwise, we can use the small RNA workbench as well. So in a small RNA workbench, we have all these integrated together. If we are not using the sRNA workbench, and if we are using individual open source tool, then after alignment, the identification of microRNA or siRNA, pi RNA using different kind of databases. So once we identify the microRNA and their respective genes, we do the differential expression analysis like DC, KEJAR, the tool remain the same, and then their putative targets and the pathway analysis using the David or the Diana microT, or there are many databases. MicroRNA DB is there. So there are so many databases we can use for the target identification and pathway analysis, and then their regulatory function of the RNA. So as a bioinformatics workflow, this is a different kind of analysis we can perform. We can perform microRNA, we can perform the gen code genes using star. So star is also a pipeline. Top hat also can be used. So because for any RNA data, we can use anything. So this is using the gen code pipeline. The outline is here for the gen code. So here we use the VGCNA and lemon tree for the co-expression analysis. So this is the extra step as compared to the previous one. So this is the comprehensive pipeline for a small RNA. We get the FASTQ raw data file. We merge the FASTQ and filters by the barcode, merge the FASTQ files, and then clip the FASTQ files, unique sequence read, map to the reference genome, and then we go and just align with the database also, only the transcriptome, no need to go for the genomic. So we take the mRNA database, RefSeq database, and then map the reads to the reference RefSeq database. Bow tie to transfer RNA, snow RNA. So these are the different kind of aligners for different kind of RNA. because different kind of RNA will have the different kind of read length. So that's why we use the different kind of RNA. So BOTI2 will use for tRNA, RNA. So there are different kind of database and other things so which can be used. For piRNA database is there, for piRNA, ensemble transcript database is there, microRNA database is there for microRNA, and then we get the count summary for each kind of microRNA, and then we categorize the RNA by category. So this is another kind of how, if we have the mix of all non-coding RNA, how to do the analysis using different kind of tools. So in these kind of tools, usually one tool will not work, will never work actually. So it depends what is our aim and objective we would like to analyze, and then we have to use the tools and softwares according to that. So further to that analysis, we can create the volcano plot showing the differentially expressed microRNA, and we can label that. We can see that this is upregulated, this is down-regulated, and each microRNA can be labeled. And this is the, for each microRNA, how they are relatedly expressed. So this is the bar plot we can create. So with this, I end my session. And before moving to the hands-on and show some tools and information, if any question is there, I would be happy to take at this time point. So I could see there is a question. Normally we do tissue RNA sequencing analysis mass. Yes. There are 2-3 objectives when we do the RNA sequencing analysis from mass RNA sequencing analysis. So we won't get, we will get just the differential gene expression or the alternating splicing information from there. But if we would like to see what clones are there, because at some time point, the tumor microenvironment is such a vast microenvironment, or any immune filtrating cells are there. You are not, so clinician is not able to treat the patient if it has the drug resistance, relapse, how you will monitor. So that is not possible with the mass RNA sequencing analysis. So that kind of information we get only from the single cell RNA-seq. But the cost is more for single cell RNA-seq as compared to RNA-seq. So single cell RNA-seq is more common from the research point of view, not in diagnostic, but probably over the period of time, single cell sequencing analysis will be preferred over RNA-seq analysis. Because still for diagnostics, when you put any test in the diagnostic, there need to be some guidelines so that every lab should follow. So still there is no guideline, because if say suppose for the same type of tumor, you map 2 individuals, it will be entirely different. So how the data will be generated, what are the guidelines for data analysis, how to report. So that guidelines are not there yet, but maybe down the line they will be there. So hope you get your answer, David. Any other question? Please, you can unmute and ask the question. So if no question is there, then R is most preferred. See, all these, when the analysis is started, so people build the packages in R. But these days, whatever you want to do in R, you can do in Python as well. And if you consider the Jupyter notebook, now there is a release that you can install R in Jupyter notebook and use it. So whatever the package you have, DC, Kedjar, Lima. You can install with Python in a Jupyter notebook and use it. Because for R, still you need a little bit of coding and the syntax. You need to understand the syntax, but in Python, it is easy to understand, even for a biologist. R is a bit tough than the Python actually. But once you know how it is, how to use it, then it is easy. See, there is no kind of book. So if you want any package or anything, so you can go to Bioconductor or CRAN repository for R. So same is for Python. You just Google it and you will get. This is how I also learned. And you can ask in your, like, if you have any doubt, you just put, there are so many forums actually for Python, for R. If you just put your question over there, you will get the answer. I get always, if I have some problem, I usually put in the forum and I get the answer. So you Google it, there are so many good videos on YouTube. You just go and watch them and learn. So I started learning from YouTube, Python and all. There are many. Probably I can send you some good links. Share your mail ID. I can share some good links with you, YouTube. So anyways, I think there are no more questions. So we will just move it, because I wanted to show you single cell, how you can generate the clusters in R, which is very, very important. Okay, so I'll just share my screen again. Just a minute. Why again it is showing? Okay. Okay. So we are here. So we have in R, there is some package and libraries. So there are some packages in R. So the main package is SWOT. So this is from the Satija lab. They have developed this for a single cell RNA-seq analysis. Apart from this, we need deployer library, we need patchwork, we need kern smooth, we need LME. So first, as a first part, we have to install the package. If the package is in CRAN, we can just type it install.packages and write the package name and then run it. And then we have to call for the library. So as a first step, the tool installation and the library, calling for the library. So which I have called up. So now I have a PBMC data. So we know that in PBMCs, we have different kind of cells. Like we have NK cells, we have CD44 cells, and so on. So we have different kind of cells. So I have downloaded this data from 10x Genomics website. So this data is freely available, or there are many data is available from SRA. So before I go into this, I will show you first how we have generated, we should generate the count matrix. So the count matrix generation is same as we do it in the mass transcriptomic data analysis. So let us go in the Galaxy, and then here I have two samples of three replicates of two samples. So one, I have the blank RNA, like a control, where I didn't give any treatment, and this is a treated samples. So there are three replicates for blank and three replicates for treatment. So if we look at the read data file, this is a FASTQ we see, and because this is a small RNA, so the read length is also small. So let us do the QC and we will see how the QC is. So let us just do the QC with FastQC. So go to FastQC and go to multiple data set. And in multiple data set, first we can select all the blank together and then execute. So this is executing. We can just go for another data set also. So go here and then select other data set. Replicate 1, 2, and 3. And then just do the QC. So let this go to QC, and I hope that most of you people know how to align this with the reference, because this is we know the reference, so it is an alignment with the reference. We can do anything. We can do high set 2, we can use top hat, and we can do any kind of alignment and generate the BAM file. So once we have the BAM file, meanwhile I'll just show you so many servers where you can do the analysis, and then we will go to R. So this is one very good website, Alona, a single cell analysis pipeline. So this runs on the web. No need of any coding or anything to download anything. So you can just upload your data as well in this. But just before uploading your data, you have to zip the folder. That is the only condition over here. There are so much of data set over here. You can download and just have a hands-on and then do your own analysis. To do your own analysis, go to upload and analyze your data. And here you can do add. So when you are here, so whatever the data set you have there, you can just browse the file. So we have some data set over here. So these are the, you can see the zipped file. And no need to make anything, just upload. This takes time. The only limitation is this takes time, and you can see that it is uploading. So let it upload. We'll give some time to upload. Yes. After once I will show you, we will share this R script with you. So this is uploading. Let it finish this. So if we look at different kind of things, this is the GSE number. You can go to GEO dataset and put this number. You can read more about it. Then quality filtering is there. Then the tab or space, how you would like to generate the data. Metrics, yes. Then the normalization is standard. You have other methods also. You can use which strategy you. So this is this SWOT. This is the basic main pipeline for single cell. So this is most popular. So we will generate from this. Then the PCA components, yes. Which species data you have, you can choose from here, mouse, human, or other. So it will cluster based on SNE instead of UMAP. You can choose UMAP also. Then the clustering, we have three algorithm over here. Leaden is most common. And then if we want to do the DE expression, so either you do the t-test or the Wilcoxon test. So usually t-test is more common. So t-test, but you can choose other things also. So percentage of expression, that also you can select. Cell type markers, you have custom, you can customize, or we have the database also. And then batch correction, whether you want batch correction or no. So once the file will be uploaded, you can do. So this is one I will just put this link also in the chat box so that you people will have the link. So then another one is this also. I will keep this. You can visit this and read more about this using this link. So this is a single cell RNA-seq workflow in Jupyter notebook. If somebody has the NVIDIA GPU card, you can use this. So this use the rapids and ScanPy for single cell analysis. So this also I will give you. So the code is available at GIT Hub. You can use it. So another is, so this is what we are doing in Galaxy. The another one is, so this, if somebody has the Windows version, you can install on Windows. This also I will put in the chat box and do the use for RNA sequencing analysis. This is free. Anyone can install on the Windows and they can install as a virtual machine also for this database. Now this is another one. This also I will tell. But here you cannot upload your data set. It is for the public data set. If you just want to have a how it performs, but you can analyze your own data set also. But there are some restrictions. So here you can select which particular human reference you would like to use it on. And from here, there are different kind of databases. So you have a Dasher data set and you have the ENCODE data set. Here already so many data set is there. You can select from here and then analyze with the Dasher. And from here also you can select something, whatever the data set you want. You can select, there are so many data sets over here. Select and then analyze with ENCODE. So I have done some analysis. So if we see here how this gives the analysis. So it gives the read versus RNA class, which RNA class will have in your read. Peaks versus RNA, what are the different peaks you will get over here. Then interactive peak browser, you will get the table over here. This is small RNA. And then you can visualize in the genome browser. UCSC genome browser, you can browse this. So you can see which kind of this is the peer ID, this is HSA, this is the microRNA, this is also microRNA specific to human. You can see all. You can see the plot. You can see the plot. You can download all the results and then you can do some other downstream analysis. If you want to make the regulatory pathways and other things, you can use this data. You can download this. So another database is there. So how it comes? So you can use everything. So plots and everything it will give, and you can download this entire data and then use some other tools to visualize. You can make it crisp. You can use for network or pathway analysis, and you can search in the data, a specific database as well. So this is this database. This also I will keep it in the chat box. You can use. Then we have seen this. Now this is another one. This also I will give you. This is very interesting, this database. So here you can keep your sequences of your microRNAs and then you can submit, upload and submit. So here you can upload your file sets, data files itself, sequencing files itself. So we will come back to this page, still it is running. This is one more web server, single cell transcriptomic annotated viewer. So here you can upload your data. You have to create your login and password over here on this web server, and you can upload your data. There are some integrated workflows are also there. From here you can just select, you can select this, you can select this and just say load selected data. And from there, then you see how beautifully you can see the clustering of the cells. So in this data set, you can see that NK cells are there, B cells are there, memory T cells are there, DC dendritic cells are there, Th1 cells are there, Th2 cells are there. So you just keep cursor here and you can see all these. So this plot is generating using the TSNE. So if you go here, expression, you can see the individual expression also. No genes are selected, just go here and then select the genes. So whatever the you want, you can select from here. So if I want immunological genes, I can just select that, this data, and then there is list. So I can just select and then go ahead from here. So here I can select the groups. If I want tissue, I want the which cells I want to see, clusters, DC, or I want average expression or the rank or enrichment score. I can just select this and plot. So this is working. See now this that has given this. So this is identify all these. So this is the targeted gene. This is the microRNA. This is the sequencing and the alignment. Everything this tool gives. So till alignment, it gives everything. And then again, you can filter and do more analysis. You can download this database as well. So now it has given you the cluster expression by genes. So because we have selected the dendritic cells and the immune cells, it gives for that. Right? Cluster is dendritic cluster, enrichment is score. Distribution. So what the distribution we want? Expression, group splitting, tissue, and then go here. So it is given the violin plot. So it will show you how the genes are getting expressed. So this is the effector memory T cells with mean and everything, quartile regions also it has given. So this is your DC. This is your neutrophils. So it is clustered by your tissue because we have given the groups split by tissue. So it is blood, lymphoid. This is the normal, this is the tumor tissue. So if we go to the detection, so we want individual express. So what it is? Enrichment score, individual rank. We want individual expression and we want for the tissue. And we want top 10. So you can do either top 30 or top 10 and plot. And then it will plot the matrix for you for top 10 genes. How it is plotted. So similarity, we want individual expression, we want by tissue, we want for top 10 and plot. So now this is another server where you can do So you can load your data set, pre-processing. So it will pre-process the samples. It will give you the count, matrix count, everything it will give you. Then it will give you the cell plots. So what is the population it has. It will do the clustering. And this particular tool use the UMAP clustering. Then the feature plots, what are the different features are there, and the violin plot. And then you can download even the results from this server. Another is this server. Here you can upload the samples. I'll just show you. We can do the expression table. So here we have the expression. So this takes the CSV file actually. So it is uploading. So if you do not have the metadata, then you can just select this no metadata and then process. But it is better to give the metadata. Then you will get more better visualization of your data. So when you have your count matrix or the differential expression, then store that values in the CSV format and upload. So then browse your metadata. So metadata. And then add data set. So loading the data. So when it loaded the data, you can see what it is. It has given. So this is the genes. In your y-axis, in the rows, your genes are there. In the columns, your samples are there. Metadata table, if you look. So here is the data information, what it is. And then we can just submit. So now you can see that it has so batch effect type. So whatever we want, we can put that. So I'm just running as a default settings, but you can play around. So cell labels from metadata. So you can see that it has clustered the cells for different samples. And it also has used the TSNE. This is your principal component analysis, and this is your cell clustering. Then submit. So now you can see that it has corrected the batch effect. Then again submit, and you can download even normalized count and then use it in R. So this is the log expression level. And submit. So how it has done the normalization. Dispersion. So this is the dispersion graph. Now you can see any clustering method you want, you can choose. So you can use the Euclidean, you can use TSNE, you can use hierarchical clustering, you can use with the TSNE, you can use non-negative method, but Euclidean k-means is very common. And whatever the clustering, how many clusters you would like to generate, you can mention here. So minimum is 3, maximum you can increase 4 or 5, whatever. So I'm just running on the default. But you can change these options and run. We'll just see whether it has come or not. There is some problem, I think, on the server itself. Anyways, we will see this because this is not that important. This is a normal routine fast QC, and then just create the count matrix using the high set 2 or top hat or anything. So once you have the count matrix, so because I would like to show you how you can do this analysis. So I have saved. First you go from here, session, and then set the working directory and choose the location wherever your files are there. So then I create the data from the count matrix. So I will run this. You can see. So I'll just clean this first, so that it will be easy for you to see. Okay, anyways, it will be done. So as a first part, we have to read our data. So in SWOT, there is a operator called read 10x. So because this data is generated from 10x, so just we have given that and created a matrix. So it is a PMC data. You can see how many elements are there in this. So these many data points are there. So if we look at what is the data in it, how the data looks, just run this and it will show you the data over here. So this is the matrix. So now if we go and do the PMC, it is still running. So you can see that this is the data. This is the data matrix from each cell. Then in this, we are doing the counting. So when you can see this, it means that it is running. When it will return into the greater than sign, it means that it has run. The command has run completely. So now it has written, it means that that command has run. So if I would like to see what is PBMC, so it has 13,714 features across 2,700 samples within one assay. So if I want to see what are these, so these are the markers for me. So I would like to see this. So now you can see these are the read counts for these genes. So what is the size of this data? It will tell me. Because we have to do the normalization and dimensionality reduction. So with this, it will create a matrix. So still it is running because the data is huge. So this is the limitation of R. It takes a bit more time to run. Same thing I can do in Python as well. Yes, you can do, but this is then you need to change this code because the same code will not work as such in Python. But if you will install this R in Python, then you can use this. So there are other libraries which you can use in Python. So now it is run. So if I see the size of my dense, then it is this much of byte. You can see that how big it is. So if I want to see this size, see this much big is the matrix. So I will do the reduction. So and I will see the what is the percentage. I will divide the dense size divided by sparse size. So then this is only 3.7 bytes. So here what I am doing, I am just creating the percentage of features. And then I will plot as a violin plot. So you can see that this is the violin plot for the feature count RNA and then the PBMCs. So here I am doing in these two, plot 1 and plot 2, I am just doing the normalization and then creating a plot together. So now you can see the identity of the cells. Then the subset of the PBMCs. So we have collected the subset of the PBMC and then doing the normalization. So this is the normalization. Then again the normalized data PBMC, just I'm looking at it, how it is. Then this is the find variable features. So what are the variable features in this? Then top 10 features. So here you will get that these are my top 10 features here. So it will create here. Then my plot 1. And then when I will create the plot 1 and 2, we can see over here. So we can see the PCA over here. So now all the genes in my rows, and then I will do the scale data. So it is scaling my data. Run PCA on the scaled data. So then you can see the cluster. You can see that these many clusters will be there for all this. And then I am printing the PCA. So now you can see the PCA, principal component analysis. So you can. So this is the just different styles of plotting the PCA. So you can see the heat map. So now if we zoom this, you can see clearly. So here it is the each principal component analysis cell wise it has done, sample wise and the cell wise. So you can see how it has created. So here the genes are there, here the samples are there, the cells. So this will take time because the number of replicates is 100, and then it will count. It will take the subset of from 100 replicates. For each cell, 100 replicates it will create. So this will take around 6 minutes to finish it off. So let us see what is happening over here. We'll just run again. Let it run this. Just go back. So this is running. And see that these 3 clusters. If you change the cluster and these other things, then it will generate the heat map. So like that here also you can upload your data and do all these kind of analysis. So the advantage on web servers that you do not need really a good coding, but just you need to understand the parameters and how they work. So now it is run. So if we look at here, so we can see that everything is fine. We can look at the QC, right? So the adapters are not there. We do not really need to do anything. So all good, just we have to check here the adapters because they are very short read, maybe 50, so 40, so what is the length? 12 to 50, that's it. So it is less than like up to 50 base pairs. So really adapters will not be there because adapters itself is a 20, 23 base pairs. So usually in small RNA, we don't see adapters much, but just for the safer side, we have to do. So this is all good to go, right? Just now we can go to high set 2. You use high set 2 because in this that top hat and that cufflinks are not there. So you can't use really that. So use high set 2 if you are using the Galaxy. So use a built-in genome. So we can go to reference. So there will be a reference. Human HG19, use this. And then this is my single end library. So I'll just use a single end and then use this. Then advanced option. That's it, nothing, and just have to execute this. And then it will give me the BAM file. So once we have this BAM file, we can get the count using any string tie or anything we can use and get the counts. And then we will get for each sample, we will get the count matrix. Okay, let us see whether it is done. Yeah, it is done. And see how it is. Still 53 seconds are there to finish it off. Yeah, so it is done. So now you can see that it is created the principal component with the p-value. So this is how it has done the dimensionality reduction for whatever the 100 replicates it created. So then find the neighbors. So it has found the neighbors. Then what are the indents for that? Then run map. So UMAP it has created. So we have seen that we have two types of clustering. One is using the TSNE and another is the UMAP. So this particular pipeline used the UMAP. So now it has made the cluster. So this is a PBMC data. So at this point, it didn't label this, but it has made the cluster. It had identified the cell types. So if we want, we can save this plot in the RDS. So this RDS we can visualize in our loop browser, which is from the 10x genomic. I will show you how we can see the visualization, the cell type fraction, and do some more analysis and the visualization of data there in loop browser. So this is the Lima package for differential gene expression. So there are three most commonly used packages for differentially expressed gene analysis. One is HR, DESeq or DESeq2, and Lima. So here in this pipeline, we use Lima. But for the same pipeline, we can use DESeq or HR also. So, Lima is a bioconductor package. So if we want to install any bioconductor package, so this is the code for that. And then call for the library. I'm not running because I have run once. So then the cluster in one. So the whatever the cluster one is there. So red color. So this is my cluster one, 0. And then brown is cluster 1, 2. So this color will give me which cluster it is. So there are total 9 clusters here, 0 to 8. So we are just seeing the what markers and creating the tables over here. So you can see that this is the cluster 1. So these are the cluster 1. And when I'm saying the n is equal to 5, so from each cluster, I'm just taking the 5 top differentially expressed gene. So cluster 5, cluster 6. So I'm just randomly picking few clusters, but you can run the same code for all the cluster like 1, 2, 3, 4, 5, 6, 7, 8. Then I am finding the markers. So in each cluster, it will see the marker. So 0 to 8 in each cluster, it will show. So what are the markers present in each cluster? So these are the different kind of cells. And you can see that in 8 cluster, very small fraction is there. Yeah. So sometime it will show some error because the data is very huge. It won't be able to compute and show over here, but it will run at the back end, and when we will see further, it will generate. And now you can see, so you can see the difference between the previously generated violin plot. So this is like from each cluster, this is the genes. So this is the gene name, and this is the cluster, this is the identity in each cluster, how it is. So whether this particular gene is there in each cluster. So this is the 0 to 8 cluster. This is the gene. So if we look at this gene, so this is the highly expressed in 4th cluster. And if we see here, PF4, it is in 8th cluster. So like that, we can see in which cluster which gene is highly expressed. And the feature plot. So if I do the zoom in, so then you can see the U map and this. So it has made the cluster for the each gene in that cluster. So this will show because it will not fit in this window, but it will run. At the back end, it will run. So this is top 5, top 2, that's it. And it has created the. So now we see that new cluster we are creating for NAV CD4, memory CD4, CD4 mono, B CD82, CD8T, FGFR, NK, dendritic cell platelets. And now see, now we have created the new clusters with the labels. So now we would like to know each cluster, which cells are clustered together. So this is the platelet, this is the FGR plus mono, dendritic cells, memory cells, CD8 T cells. So this is labeled now. So we have saved that plot with the labels. So now we are plotting the top 10. So for each group, top 10 genes we are plotting. So now you can see the finally we have validated that yes, these are the our cell cluster and identify the cells. So this pipeline is for the identification of different cell types from the each sample. Right. So let us see what is happening here. Okay. So now this I said to this BAM file is being created. So if we will look at it. So this is kind of a normal BAM file. Right. So if we want, we can. So same, if somebody would like to use string tie, they can use string tie also. But we want to count now. Count. So HTC count align reads in a BAM file that overlap features in a GFF file. So we can just count this. So already it is there. File. And then we can execute. So for that we need to have the GTF file. So how we can do the GTF file? So we need to download the GTF file, and I think it will be very easy, very heavy GTF file, but I will show you how you can download the GTF file and import. So usually like go to iGenome, Illumina, go here, and then from here you can, because we have used GRCH38, so from here you can download this GRCH38, because this is the entire folder. If I would like to download this entire folder, it will take time and I cannot download this. So in this you will get all the indexes together, you will get your reference file, and you will get the GTF file. So once you get your GTF file from here, you can upload this GTF file here, and then you can use this for the feature count. There are other methods also for the feature count. Then it is not coming. So once you have your reference and GTF, if it is not available in Galaxy over here, you can upload here and then use it. So then you do not need to select from here, just choose from the upload, and then you count the matrix. Or otherwise, I showed you something where you can, I'll show you, you can just download this BAM file, and then directly you can upload here this BAM file. I'll just show you. There is one browser, you can just upload your directly BAM file, it will give you.
Yeah. So I think from when it comes to the future, there's a different section. I think the specifically of those section or the instrument which we are utilizing with that. We can come up with some information.
There is one browser, actually you can use directly BAM file over there. This I have given you already, this link. Yeah, here you can put your input type. So here you can put your mapped BAM file, right? And you can download the BAM file from here. How you will download? Just go here, and from here you can save this BAM file. So download data set, and at the same time download the index also. So meanwhile, if anybody has any question, please unmute and ask question. Meanwhile, I'll show you how you can upload your BAM file over here. Yeah. So this is the BAM file, and analyze your own BAM file. So how it will take, it will calculate for each chromosome from your BAM file. So this is running. It will take some time. Meanwhile, it is running. You can ask questions. Here just you have to upload your BAM file. You do not need to do anything, and it will, from your BAM file, it will analyze the data. So it is loading. It will load your peak files. You can see in the browser. So everything you can download these results, and then you can find in the databases, respective databases as well. So I can show you how you can map, enrich your genes or small RNA. So there is a web browser called gprofiler. Go here. This is not only for like, this is just, you have to put your some genes over here. Say if I put NPP1, HPRT1, if I put some other genes like BCHE. So just put your here and just select your organism from here and run the query. And then it will give you the info over here. It will classify your genes for your molecular function, biological function, cellular function, pathways. So you can see that it is a purine metabolism pathway, riboflavin metabolism pathway. So this is nucleoside pathway. So everything it will give you. So reactome pathway, then a transcription factor, microRNA. It will classify your genes and enrich. And then you can download this data as well. So this is your, it will give the p-value enrichment. So then these data also you can download. Everything, this is my query. So the genes which are coded by these ensemble IDs, it will give, and it will map for all these pathways and information. So this is also a very good tool, and you can export your graph. You can show the, you can get your query, what it is. You can get the link of it, and then you can just download and use for other purposes as well. So these are the good tools where you can do online analysis. You do not need to worry about how to create the pathways and all. If you want orthologs for this, you can just export to CSV. So this is your ortholog search. If you want SNP information, you will see what are the kind of variants these genes have. So, and then you can just do this analysis from here. So with this, I end this session over here, and any more questions, please ask. Yes, I'll do that. Any questions? BAM file, how I can input in R for analysis. So for a BAM in R, you need to have installed SAM tools. Then from there you can get the feature counts. So even in R also you can install all these tools, POPET, SAM tools, BAM tools, VCF tools, everything you can install in R and do it. Your alignments, your everything, a small data set you can perform in R. Even if you have a very small data set for ChIP-seq, that also can be done in R. Everything you can do in R. So after count matrix, I showed you all these steps which you have to create. So identification of your cells, then counts of what genes are there in each cluster. So that is a kind of, and then differential gene expression. Because whatever the genes you will get, each cell will not represent the genes. So those genes will be, some genes out of them will be cell specific. So based on those genes, the tool will identify the specific cell. And once those genes will be mapped, then the differential expression will be done. So if no more questions, then we will end this session over here. See you in the next sessions. Thank you. Good night. Yeah, I noted down. Thank you.